Welcome to Excellent Grades Academy. This is Dr. Byson EM. And in this lesson, we're going to look at histology, lab practical number 9, which is muscle tissue. Let's move on and let's look at the objectives of this lab. What are we looking at? So the learning objectives after this lab, the student should be able, number 1, to describe the light and electron microscopic appearance of smooth muscle, skeletal muscle, and cardiac muscles. We're looking at these 3 muscles in this lab and how their histology is. Number 2, you distinguish the 3 types of muscle by their light microscopic appearance. Last but not the least, which is the third, is define the term hyperplasia and hypertrophy. Okay, so we'll start with striated skeletal muscle. And we're going to use slide 43, which is the root of the tongue, to study this striated skeletal muscle. So at low power, you identify the muscle fibers, which are the fascicles, and they are cut on in longitudinal section. At high power, you note the regular cross striations and identify the A and the I band myofibres, which appear as fine longitudinal striations. So you make the drawing at both low power and at high power. So let's look at the slides that we're going to look at. So this is the slide at low power. Okay, so in this slide at low power, you're able to see this is the root of the tongue. And at the root of the tongue here, you're able to see this. This is a longitudinal muscle fiber, which is a fascicle, and this is a longitudinal muscle fiber. So they are running in longitudinal section. That's how you are able to appreciate them. They're longitudinal. Okay, so these ones here that appear like this, these are cross section. These are cross section fibers. So I'm able to appreciate the muscle fascicles. Okay, so these are the muscle fascicles. The muscle fascicles. Muscle fascicles. Now, these muscle fascicles are bands of muscles that are covered by connective tissue that is called the perimysium. And the muscle fascicles contain individual muscle fibers that are covered by connective tissue that is called endomysium. You're not able to see this here because this is at low power. Okay, so an individual muscle fiber is covered by an endomysium. So muscle fiber is covered by an endomysium. All right, so at low power, I just want you to draw this. Draw the muscle fibers in longitudinal section and also draw the muscle fibers in cross section. What you have here, this is the epithelial layer of the tongue, and we know that the epithelial layer of the tongue is made up of stratified squamous epithelia. Stratified squamous epithelia. So this is a stratified squamous epithelia. And you have papillae here, different types of papillae. Filiform papillae, fungiform papillae, valate papillae, and foliate papillae. But our interest is not the epithelia because we've already done this lab. Our interests are the muscles, which are these muscles here and these muscles here. So just draw this section starting from here to here. From here up to here. Draw this section, show the muscles in transverse and show them in cross section. So this is at low power. At high power, they appear like this. Okay. So high power, these are the muscle fibers. So this here is a muscle fascicle in longitudinal section. Even this one here is in longitudinal section. So draw these ones, longitudinal section. Now what you notice in these muscles is that they have a lot of nucleus. So there's a nucleus here, a nucleus here, a nucleus here, a nuclear here. They've got a lot of nucleus. That's what you notice in these muscles. They have a lot of nucleus. So we say that skeletal muscle is multinucleated. It has a lot of nucleus. Let me just change the color. It has a lot of nucleus. A lot of nucleus. Okay. So draw the longitudinal skeletal muscle fibers. This is a cross-section muscle fiber. This is a cross-section muscle fiber. This is a cross-section muscle fiber. What you see here, okay, just a minute. What you see here, this is just connective tissue surrounding the muscle. And then this is a gland. This is a lingual gland. Okay. This is a lingual gland. The lingual gland here. So you can draw the lingual gland, but our interest is in the muscle fibers. Okay. So draw longitudinal muscle fibers and also cross-section muscle fibers. So you draw this at high power and you are done with skeletal muscle. Let's move on to cardiac muscle. So under cardiac muscle, at low power you observe that the myocardium forms branching continuous bulk, and they are not grouped into bundles or fasciculi. So at high power you note that the cardiac myocytes have cross striations and that they have a single central nucleus. You also find the intercalated discs which separate the individual muscle cells in cardiac muscle. Okay. And you also note the connective tissue that the cardiac muscle has. The connective tissue that the cardiac muscle has. Very important. So you make the drawing at low power and at high power. And you also note the PURKINJE fibers. PURKINJE fibers are what we call specialized muscle fibers that are able to conduct current, the conducting systems. So they are able to carry action potentials to all the parts of the heart, especially the ventricles. Okay, so draw at low power and at high power. So this is at low power cardiac muscle. What you see here, this is cardiac muscle, and then it becomes branched. So one characteristic of cardiac muscle is that it is branched. It is branched, and you can see each cell has got a single nucleus. So there's a nucleus here, there's a nucleus here, there's a nucleus here. So cardiac muscle is highly branched, very highly branched. This is cardiac muscle. What I'm drawing here is cardiac muscle. So here is cardiac muscle, highly branched. So between the muscle here is connective tissue. Okay. So what is separating the muscle fiber here and this muscle fiber here is this part here, which is connective tissue. So make sure you draw. This here is a nucleus. Okay. The nucleus of a single muscle fiber. The part that you see here that is dark, this is what we call the PURKINJE fibers. Okay. These are PURKINJE fibers. So when you draw this, include it as well at low power. These are PURKINJE fibers. So draw here, show the branched cardiac muscle, the connective tissue, and the single nucleus in the cardiac muscle, and also show the PURKINJE fibers. This is at high power. So at high power, you're able to see very clearly how the cardiac muscle here become branched. So they are very highly branched, very highly branched. So it comes here, forms a branch like this. Cardiac muscle work in a syncytium, meaning that they contract at the same time. And as you can see here, it has a single nucleus. Sometimes they can be binucleated, but most muscle fibers just have a single nucleus. This one has a single nucleus here, this one a single nucleus here, this one a single nucleus here. So hallmark, I'm repeating it. Cardiac muscle is highly branched and it has, most of the times, a single nucleus, although it can be binucleate sometimes. So cardiac muscle is not multinucleated like skeletal muscle. It is just mononucleated, but sometimes it can have two nucleus per muscle fiber, which is binucleated. It also has striations. What are striations? You're able to see inside the muscle fibers here as if they are parallel lines there. So like skeletal muscle, cardiac muscle has got striations. It has cross striations. Now, one hallmark of cardiac muscle is that the individual muscles are separated from one another by a structure that we call the intercalated discs. So I added this slide here to just show you. So this is one cardiac muscle fiber. It ends here. There's a single nucleus. It is separated from the next cardiac muscle here by this disc. This disc is what we call an intercalated disc. So know that each cardiac muscle is separated from the adjacent cardiac muscle by an intercalated disc. So there is a muscle here. Okay. Let me pick another one. So there is a muscle here. It ends here up to there, as you can see, and it only has one nucleus here. That's why we say cardiac muscle fibers are mononucleated. They are mononucleated. You find that here this one is branched. It ends here where the intercalated disc is, goes up to here where the intercalated disc is. Then it goes up to here, there's an intercalated disc here, and then again it forms a branch. So the way I'm drawing it like this, there's an intercalated disc. This is how you're supposed to draw it. There's an intercalated disc. There's another one here, and then there's a nucleus here. There's a nucleus here. Actually, two nucleuses here. So cardiac muscle is highly branched, has intercalated discs that separates individual muscle fibers from adjacent muscle fibers. Okay, so that's cardiac muscle. And the spaces between muscle fibers, this is just connective tissue. So draw it like this and make sure you label all of these parts, both at high power and at low power. Now let's look at Purkinje fibers. So this part here are Purkinje fibers. We said Purkinje fibers are simply cardiac muscle fibers, but they are specialized and they are used in the conduction of nerve impulses. So it will take, the Purkinje fibers will take their electrical impulses from the interventricular node to the ventricles. Okay. So as you can see, these are Purkinje fibers. They're parallel like this, and they also contain nucleus. There's a nucleus here, nucleus here, nucleus here, nucleus here, nucleus. You can see that the Purkinje fibers are thinner than the muscle cardiac fibers. These are thick. As you can see, this is thick, but the Purkinje fibers are very thin. Let's look at them. So this is at high power. So make sure you draw this region of Purkinje fibers and you label them as Purkinje fibers. Of course, you add a few cardiac muscle fibers here as you draw. Let's look at smooth muscles. So the hallmark of smooth muscles is that they are spindle-shaped. A term that is used is fusiform. They are spindle-shaped individual muscle fibers, and they are not striated. They are mononucleated, meaning that they have only one nucleus, and the nucleus is centrally located in the cytoplasm at the widest part of the cell. The nucleus are sausage-shaped with rounded ends. So you sketch it at high power. So this is at low power. Here you just observe. Okay, if this is not clear, so you just observe. Just observe this slide. Don't draw it. And then at high power, they look like this. But here the fusiform shape is not appreciated, so I added this slide here. So this is the smooth muscle fiber. It is fusiform shaped, and this is the nucleus. So the nucleus is at the center of the cell, and it is sausage shaped. There's another one here, and it has a nucleus here. So how I'm drawing it here, that's how you draw it. There's another one here. There's a nucleus here. So all of the smooth muscle fibers have got one nucleus. They're mononucleated. Okay, so all of these. So this slide was taken from the colon. That's where smooth muscles are found. So these are smooth muscle fibers. They're fusiform and they are mononucleated. Okay, so that's about smooth muscle. Very, very easy. So we've looked at skeletal muscles, which are striated and they are multinucleated. And then we looked at cardiac muscle, which is striated as well, is mostly mononucleated but can be binucleated, is highly branched, works as a syncytium, and has got intercalated discs. Smooth muscles studied from the colon have got a fusiform, okay, fusiform shaped, meaning that they are spindle shaped. They are spindle shaped. They're spindle shaped, and they've got a central nucleus. Central nucleus. Just one nucleus. So meaning that they are mononucleated. They are mononucleated. This is a teased smooth muscle slide. Teased smooth muscle slide. What does this mean? Teased. Teased smooth muscle simply means that the connective tissue has been removed. Okay? It has been removed, and now you only have muscle fibers. Okay, so these are just muscle fibers of smooth muscle. The connective tissue has been moved, so you're only able to appreciate the muscle fibers. So just draw this the way it is, and just label it as smooth muscle, teased muscle fibers. Teased muscle fibers. They're very visible. The reason why this is done is to show you that this is how the fibers run in a smooth muscle. Okay. To tease is simply to make straight, to comb, to make clear so that you can appreciate. So these are just teased smooth muscle fibers. Make a drawing and label it. Okay, so this was the last slide on smooth muscle and on the histology of muscles. From here, you're supposed to do the exercise. What are sarcomeres? A sarcomere is a functional unit. Okay? So this is just a functional unit of a muscle fiber. Muscle fiber. So you're supposed to know all the parts of the sarcomeres. So you draw a well-labeled diagram to demonstrate a sarcomere. I'm going to make a video that will answer this slide. What happens to the A band, I band, and the H band of sarcomeres during contraction? What happens to this? So the answer here is that they shorten during contraction. They become short. They reduce in length. They reduce in length. Okay, there's supposed to be an N here. They're supposed to reduce in length. What is the significance of the transverse T-tubules in muscle fibers? So the T-tubules, these ones, they conduct an action potential. Conduct an action potential. And that action potential leads, so which leads to release of calcium. Release of calcium. You know, calcium is the one that is used in muscle contraction. So it leads to release of calcium from the sarcoplasmic reticulum. Number 4, why are intercalated discs unique to cardiac muscles? Why are they unique to cardiac muscles? Because cardiac muscles are continuous, and they function as a syncytium. So to demarcate or to separate muscle fibers. That's what intercalated discs are there. Number 5, what is the significance of satellite cells in skeletal muscle? All right, so this was the end of the presentation. I'll see you in the next video when we'll be doing nerve tissue. Don't forget to subscribe, and we offer lessons on this number on WhatsApp. So if you're interested in registering for class in either anatomy, physiology, microbiology, organic chemistry, you can contact us on 0975 497790. Okay, so the country code is +260. Okay, I'll see you in the next slide. Study hard.